Hello everyone, welcome to Pulmonary Physiology. This lecture is taken out of Chapter 2 of our textbook. The title of that chapter is actually Cardiac and Pulmonary Physiology. I've posted a separate video for cardiac anatomy and physiology that is from an anatomy and physiology class that I take. I just think it's more detailed and that particular text approaches it in a more useful way. So don't worry, there's no need for you to obtain and read that textbook. Anything I might ask will be from that lecture. So no need to read anything for that. If you do feel a need to review pulmonary physiology, you would find that in Chapter 2. But again, really, I'm going to be trying to take my questions directly out of the lecture. So we'll start off with pulmonary functions. Now, we can't really rank these. For the normal functioning of the human organism, all of these functions need to be working correctly. So don't read into the order necessarily, but I think it's easy to say that gas exchange is a primary function of our pulmonary system. So this gas exchange occurs between the environment, the blood within the capillaries within the lung, and then the tissues where oxygen is delivered and carbon dioxide is picked up. So that's part of a longer process that we'll be talking about over this lecture, but gas exchange is certainly an important function. One of the reasons why that is that without oxygen, we're not capable of making ATP very efficiently. With a single molecule of oxygen, or in the presence of oxygen, I should say, a single molecule of glucose is capable of producing 36 ATP through a process called aerobic respiration, which I'm sure you guys have all heard about. Without that oxygen, a single molecule of glucose can only yield 2 ATP. So that's a huge difference in efficiency. So oxygen greatly enables ATP production. In an anaerobic environment, we can't function for very long. We have to stop exercise pretty quickly once we've exceeded our oxygen capacity, and this is why. Also, the pulmonary system enables pH regulation, and that's because CO2 is one of the expelling CO2 is one of the easiest ways to raise our blood's pH. The primary acid in our blood, carbonic acid, is really volatile. So what that means is it can move from liquid to gaseous form very easily. And so in the lungs, CO2 can be released from that compound, and acid can be essentially exhaled into the atmosphere, and our pH goes up, becomes more basic. So the lungs play a very important role in regulating blood pH. They also play an important role in temperature regulation. Just like we sweat on our skin, we can release warm moisture from our lungs as well to drop our temperature. There are also a lot of toxic substances that can be metabolized in the lungs. And there are also, it's not on here, but there are also some important substances that can be made in our lungs or activated in our lungs. I'm thinking of angiotensin converting enzyme, for example, which plays an important role in vasoconstriction and water retention. So our lungs do a lot more than just ventilation. They do all of these things. Speaking of ventilation, this is one of the necessary components for gas exchange. We need two things, actually, and that's on the next slide, but we need ventilation and perfusion. Ventilation is often mistakenly called respiration. It's not technically accurate. Ventilation is the physical movements that we make, the contraction of the diaphragm, of the external intercostals, to expand our thoracic cavity and take in air, and then release those muscles so that thoracic volume decreases and air is exhaled out of our mouth. That's ventilation. It's the mechanical events of breathing. So there's a few different terms and values we need to identify in reference to ventilation. The first is ventilatory rate. This is definitely an anatomist's or physiologist's term, ventilation rate. In the clinical environment, you will typically see this be called respiration rate. Anyway, about 10 to 15 breaths per minute at rest is typical. You'll see different reference ranges in different places, but 10 to 15, that's where most healthy folks reside. And for each breath, there's about a 350 to 500 millimeter inhalation or exhalation of air. So we call that the tidal volume, the normal amount of air in a normal inspiration or expiration. So you can have what's called a tidal inspiration or a tidal expiration. That's just normal breathing, how much air is going in and out. You can multiply those two metrics to get something called minute ventilation, or sometimes it's called effective ventilation. It's just tidal volume times respiratory rate. This is the amount of gas exchanged in 1 minute. And so, just to get an idea of how much this can vary, the normal minute ventilation would be 5 liters per minute. That's kind of an average. It can go up to as high as 125 liters per minute with intense exercise. This stuff is pretty big on boards, if I'm being fully honest here. So we have a lot of different lung volume metrics. These are all acronyms, and I'll tell you what each of them are. We've already talked a bit about tidal volume. So on the graph right here, you see it here labeled 500 milliliters. So this would be a tidal expiration, a tidal inspiration, a tidal expiration, a tidal inspiration. That's just normal breathing. Inspiratory reserve volume, you see this arrow here that indicates that we have this ability to take a deeper breath, a deeper inspiration than just a tidal inspiration. So if you imagine taking a normal, and I got to relax here. If you imagine taking a normal breath in, not a deep breath, just a normal breath, you can still breathe in a lot more than that. You still have a lot more room for inspiration. So that reserve beyond a tidal inspiration is called inspiratory reserve volume. Inspiratory reserve volume. And then all the spellings in your book, if you guys are having a trouble with spelling here, actually think, I'll give you a page number. Now I have the 4th edition, so your page numbers might be slightly different than mine, but so I am on page, looks like there's a diagram on page 24 that tells you what all of these stand for. So just like we have an inspiratory reserve volume, we have an expiratory reserve volume. After a normal tidal expiration or exhalation, you still have more room to breathe air out. Sorry, that might be triggering in your headphones. I'm sorry. But if you contract your abdomen and some other muscles, you can force out more air after a normal tidal expiration. Sorry if there's some background noise. Then we have something called residual volume. This is the amount of air left in your lungs after expiratory reserve volume or after a forceful expiration. There's always going to be some air in your lungs. You can't collapse them completely, nor would you want to, right? So that's called the residual volume. Then we have something called the inspiratory capacity. Inspiratory capacity is almost I don't even want to make that comparison. So after you have a normal tidal expiration, if you were to take as deep a breath as possible, so if you're kind of asked to hold your breath, all of a sudden you just take a deep breath of air in, right? And hold your breath. That's sort of your inspiratory capacity. How much air can you inhale after tidal expiration? So I don't believe it's on our graph here, but it would be from right here all the way to maximal inhalation. Then we have something called functional residual volume or functional residual capacity. FRC is actually more common. I'm not sure why I put FRV, but FRC represents the amount of air left in your lungs after a tidal expiration, period. So including residual volume. Remember, expiratory reserve volume is how much you can exhale after a tidal expiration, right here. Functional residual capacity is how much you can exhale after a tidal expiration plus residual capacity. So how much air is in your lungs after a tidal expiration? I'm going to pull a lot of quiz questions from this. So if you have any questions, be sure to ask me in lab or send me an email. Vital capacity, this is how much air you can exhale after a maximal inhalation. So you take a deep breath as possible and breathe out as much air as possible. Or you could just say the total amount of air in your lungs minus residual volume. And then total lung capacity is the amount of air in your lungs after a maximal inhalation. So this would be the biggest volume always. There's a lot of algebraic equations that can be done here. I don't want to beat you guys up too much on a quiz, but you know, prepare yourself for that possibility. TLC minus RV, what would that equal? Vital capacity, right? Total lung capacity minus residual volume equals vital capacity. So there is some potential for that. So it might be a good idea to look up and write out the definitions of each of these from your textbook and almost approach them, you know, from like a flashcard perspective, if you like, and just know what each of these are conceptually. Don't necessarily just have their definitions memorized, but understand, you know, TV, I'm trying to think of a good example of another one of those things. FRC, functional residual capacity, minus residual volume equals ERV. And you can just know that by understanding what each of these are. So I'll maybe try to only ask like one of those questions. For the most part, I'll just say the amount of air that you can exhale after a normal tidal expiration is what? It's expiratory reserve volume. Or expiratory reserve volume. All right. How do we control ventilation? Well, there are different types of ventilation, as you might guess. Most times you're not even thinking about breathing. It just seems to be regulated automatically. And that is true. There is an automatic component to ventilation regulation. That type of breathing is regulated by the medulla and the pons. The muscles of inspiration are actually innervated by centers within your medulla and pons, and that they can influence your breathing rate based on all sorts of variables. The acidity of your blood, the oxygen demand, the temperature, sympathetic activity. That's all subconscious, unconscious. You can, however, of course, hold your breath or force yourself to take a deep breath. That's a voluntary movement. And we know that voluntary movements are initiated by the motor cortices, the primary motor cortex, the premotor cortex, etc. And so voluntary inhalation and exhalation is performed by, or is initiated by the motor cortex. There's also some hypothalamic and limbic influence. I'll just read a section from your book here. Sensations of pain and alterations in emotion alter ventilation through input coming to the brainstem from the limbic system and hypothalamus. So what this is saying is that there's an afferent component, afferent, afferent, meaning a sensation component to breathing regulation. It's not just hypothalamic and limbic systems, but forgive me, I'll continue a little bit about the hypothalamus and limbic system. For example, anxiety triggers hyperventilation, which equals a reduction in carbon dioxide levels in the blood. As the rate of carbon dioxide elimination out of the lungs exceeds the rate of carbon dioxide production in the body. So we all know that to be true. If we've ever gotten nervous or had a panic attack or something, you hyperventilate, you ventilate rapidly. A lot of this decision making, I guess, of your medulla and your pons, as far as how your breathing is regulated, is dictated by afferent stimuli, like what I just mentioned, anxiety. But it could also be chemoreceptors, chemical receptors measuring the acidity of your blood. If your blood is becoming too acidic, you'll begin to breathe more rapidly to exhale carbon dioxide. You also have stretch and pressure receptors in your lungs themselves that regulate breathing. And to me, most interestingly, is joint and muscle receptors. So try to guess, why would joint and muscle receptors influence our breathing? And what's interesting is they influence them to a great degree. This really is how our body knows that we're exercising. Big changes in joint tension and stretch and muscle contraction. So this type of afferent stimuli triggers us to breathe more rapidly. We can double our breathing rate just from beginning to exercise. This happens much more quickly than could be explained by the change of blood acidity that occurs due to exercise. So this is an immediate response to exercise. So let's talk a little bit about the mechanics of breathing. So airflow through our respiratory system is a result of changing pressure gradients. If you picture this system as a tube, which it kind of is, it's just the diameter changes quite a bit. Here's one end of a gradient, here's another. Airflow direction is dictated by the pressure gradient. If the pressure is greater out here than down here, then inspiration occurs. Inhalation. If the opposite is true, exhalation or expiration occurs. So as a result of pressure gradients, the producers of those pressure gradients are primarily the diaphragm and the external intercostal muscles. These two, or this muscle and this group of muscles, is able to increase the volume of the thoracic cavity pretty dramatically. And this causes inhalation. This can actually be explained by something called Boyle's law, which states that the pressure of a given quantity of gas is inversely proportional to its volume. So during inspiration, an increase in lung volume within the thoracic cavity decreases pressure below atmospheric levels. So that's a negative intrapulmonary pressure. This difference in pressure between the atmosphere and the lungs facilitates the flow of air into the lungs to normalize the pressure differences. So by creating pressure gradients, we can cause airflow. And that's done primarily by the diaphragm and external intercostals. When these muscles relax, just the pressure of the weight of our musculature and bones and the viscera pushing the diaphragm back up returns the thoracic cavity to its resting size, which causes exhalation in the process. There's another really important pressure that accounts for this working. And so I need to tell you quickly just about the visceral and parietal pleura. We may have mentioned this in medical screening to a few groups, I'm not sure, but visceral pleura is a simple epithelial membrane that's like gift wrap on all of our organs. Visceral pleura. A visceral, well, depending on what organ it is, it's called something different, but on the lungs, it's called visceral pleura. So our lungs are wrapped in this stuff like a little gift. And then if you guys know what wallpaper is, the thoracic cavity is lined with like a wallpaper called parietal pleura. So these two membranes are in contact with one another, but there's a thin sheet or a thin little compartment of fluid, very thin. And so what happens is we have two forces working against each other at these membranes. We have the weight of the lungs and their tendency to want to collapse, pulling the visceral pleura away from the parietal pleura. And then we have the forces of inspiration, the external intercostals and the diaphragm pulling in the other direction. So this fluid compartment in between the two membranes has a negative pressure. And so that causes the lungs essentially to stick to the walls of their compartment, the thoracic cavity. So any change in thoracic cavity volume equals a change in lung volume. And so if you know anything about a pneumothorax, that's like if someone, it's kind of horrific to think about, but receives an injury that pierces one of these membranes, like a broken rib or a stab wound or something, that lung typically will collapse, and it needs to be reinflated once that wound is healed. So it's that pressure is called intrapleural pressure. So ventilation works because of pressure gradients created by the diaphragm and external intercostals and intrapleural pressure. Now that was just ventilation. That's kind of one half of breathing and gas exchange. The other half is called respiration. And respiration relies on two things, ventilation and perfusion. So we already talked about ventilation, and now we'll talk about perfusion. But first of all, respiration is gas exchange. It is not technically the same as breathing, although it's usually used synonymously with breathing. It doesn't mean the same thing. It means that gas is going from the alveolus to the capillary and vice versa. Just in case I'm getting ahead of myself, let me explain what this picture is showing us. So this is a single alveolus. This is a little single cell wall sphere. It's sort of the terminal end of our airways. So when we take a deep breath, this is where the air ends up in the alveolus. This is also the site of respiration or gas exchange. And there is a pressure gradient of oxygen and carbon dioxide. When we take a deep breath in, there's a higher partial pressure of oxygen, 104 millimeters mercury, in the alveolus than in the capillary, 40 millimeters mercury. So knowing what we know about pressure gradients, what direction is this oxygen going to go? It's going to go into our blood. And that's exactly what our lungs do. They allow for oxygen to be transported into our blood, into our capillary. An opposite, although not as dramatic, pressure gradient exists between CO2. There's much more there's 5 millimeters mercury more partial pressure of carbon dioxide in the blood than in the alveolus. So carbon dioxide diffuses and leaves blood and enters the alveolus where it's exhaled. So gas exchange has two requirements. Ventilation, meaning that there's air going in and out replenishing this gradient, and diffusion. And diffusion is caused by pressure gradients, the pressure gradients that we explained here. Oxygen diffuses into the blood, carbon dioxide diffuses out. So ventilation allows for that one half of the pressure gradient, but we also need perfusion, which is our last thing here, also referred to as Q. We need blood flow through the lungs, maintaining this same pressure gradient. So we need ventilation and we need diffusion, and perfusion is what allows for that. Ventilation and perfusion allow for this diffusion to take place. Sorry. So, so I think that's it for respiration. Just take a little pause here. The efficiency of ventilation and perfusion is increased when they are matched. What that means is, depending on the position you're laying in, some parts of your lungs may have great ventilation but poor perfusion. For example, sitting, the top of your lungs, the apex of your lungs, are ventilated well. They're actually quite distended and large, your alveoli. But the blood flow is not as good because of gravity. There's more blood flow in the bottom. So in sitting, the bottom portion of your lungs has a very efficient gas exchange. That is something to remember when we get to pulmonary interventions, because we can bias portions of the lung towards being more efficient depending on their position. So the more dependent positions of the lung, meaning the portions of your lung that gravity is acting on most, are going to be exposed to the most amount of blood flow and V and Q matching efficiency. So in the prone position, it'd be your ventral lung. Dorsal position would be your prone would be your posterior lung. Left sideline would be, your left lung, but specifically the lateral aspect, etc. So this is just something to remember when we get to interventions. All right, that brings us to oxygen and CO2 transport. We've talked quite a bit about how do we get oxygen into our blood and how do we get carbon dioxide out, but we haven't necessarily talked about how that is delivered, how the oxygen is delivered, and how the carbon dioxide is picked up. So in order to sort of explain that, we're going to use this oxygen saturation curve, or typically called oxyhemoglobin dissociation curve. So first let's talk about hemoglobin. So hemoglobin transports 98% of the oxygen in our body. 2% of oxygen is dissolved in plasma, but the vast majority is carried on a molecule called hemoglobin, which we talked about. Oh, actually, we did not talk about it in the heart lecture. I'll post an optional blood lecture if you want a lot more detail on blood, but it's optional. Anyway, hemoglobin has an iron atom, 4 actually per molecule, and if you know anything about what happens to iron when it's left out in the rain, it rusts. Another name for that is oxidation. And so that iron atom can do the same thing in our blood, which is why our blood kind of has this rusty color, red to it. So it's all about hemoglobin. Hemoglobin, the functional characteristics of hemoglobin make it the perfect molecule for picking up and delivering oxygen. And you might say, well, if something's really good at binding to oxygen, it doesn't seem like it'd be very good at letting go of it. And if the environmental conditions are the same, you'd be right. But fortunately, environmental conditions change depending on what part of the body you're in. So forget about left shift, normal, right shift for a second. What I want you to see is that, first of all, different pressures of oxygen result in different levels of oxygen saturation. But then the environment can shift that curve to the left or to the right. Okay, so a left shift would be caused by decreased carbon dioxide, increased pH, so decreased acidity, increased temperature, etc. In that environment, hemoglobin is really attracted to oxygen. That's increased oxygen affinity, which means that it picks up oxygen. It's not good at unloading it. So if you think about, what environment would there be the least amount of CO2, in the lungs or in the tissues? Well, where is CO2 made? It's made at the tissues, and then it's returned to the lungs where it's released. So the CO2 concentration is low in the lungs, or at least in that portion of the blood, the pulmonary circulation, which means pH is high, and which means temperature is relatively lower because there's environmental air in the lungs. So that makes oxyhemoglobin really want to pick up oxygen, and it does. It picks up oxygen. But then it's delivered to the tissues where there's more CO2. The temperature is higher, the pH is lower, and so oxygen dissociates, which is why this is called the dissociation curve, with hemoglobin, and is released into the tissues where it's needed, which is exactly what you want. And so it's the ability of hemoglobin to change its affinity with oxygen that makes it such a good oxygen transport molecule. It doesn't really move much carbon dioxide. It moves some. So about 10% is dissolved in plasma. Another probably 10 to 30% is bound to hemoglobin, but not the iron atom. It's bound to the globin proteins within the hemoglobin molecule. The majority of it is transported via a bicarbonate ion. That's carbonic acid dissolved in water, bicarbonate ions. And so it makes its way back to the lungs in this form, where it is then released through expiration. Well, first it's there's a gas exchange from the blood to the alveolus, and then it's exhaled. And then we're pretty much done. Lastly, just, we briefly mentioned this a few times, so I just want to make sure I reiterate it. The lungs are responsible for regulating blood pH, also called our acid-base balance. So most metabolically produced acids are eliminated as CO2. Carbonic acid is the most common metabolic acid in our blood. It's the most common blood acid, I should say. that hydrolyzes and becomes bicarbonate ions, which CO2 can be taken off of a bicarbonate ion and exhaled. So most of our acid removal, I guess, is done in the form of exhaling CO2. Other acids are regulated by the kidneys and liver, but the lungs are a big part of maintaining this acid-base balance. So guys, thank you for listening to this lecture. If you have any questions, just be sure to pull me aside in lab or send me an email. You can even shoot me a text message. Have a good one. Bye.